Welcome to our mini-seminar series on improving the production and recovery of recombinant proteins. This lecture concerns why the medium composition used to produce a recombinant protein can mean the difference between success and failure. The focus of the talk will be on E. coli expression systems. Escherichia coli is among one of the most studied organisms with a 40-year history of being used to produce recombinant proteins. This system has several key advantages over other systems, which include a short development time, inexpensive and well-validated tools, and a low-cost production system. There are some drawbacks, however. Protein preparations can be contaminated with endotoxin, residual pieces of the lipopolysaccharide that compose the outer matrix of the bacterium that have pyrogenic properties. Care must be taken in removing endotoxin before performing any animal or human studies, as well as for certain cell-based assays. Many heterologous proteins that are highly overexpressed can accumulate as insoluble products known as inclusion bodies. Further, while some proteins may be exported to the periplasmic space, it is rare for proteins to be fully exported to the culture medium. Finally, E. coli, like all bacterial systems, does not glycosylate proteins, a post-translational modification often observed in eukaryotes. Despite these drawbacks, the E. coli system is often the first choice of many researchers for producing a recombinant protein. As one of the most highly studied and used systems for producing recombinant proteins, many of the processes which affect expression have been identified and schemes devised for optimizing and overcoming expression barriers. These schemes involve modifying the gene sequence, plasmid vector, and host before production is considered. Several modifications to the gene sequence can optimize for codon usage, eliminate inhibitory mRNA structures, modify high GC content, and eliminate regulatory motifs and repeat sequence. The choice of vector allows for the selection of plasmids with different copy numbers, promoter strengths, and other regulatory elements that modulate expression levels. Finally, different host strains, or the genetic modification of host strains, is used to mitigate specific barriers to expression and is often advisable to assess expression of a target protein in different host strains. Changes in any number of nutritional and environmental factors can influence the volumetric production levels during the fermentation and protein production phases. The influence of the culture media composition will be discussed further. Recovery of a recombinant protein is also influenced by the downstream processes after the production by fermentation. The approaches used for extracting, purifying, and stabilizing the protein all influence recovery and are the subject of another mini-seminar. The upstream processes, however, will affect the downstream processes, so care should be taken when choosing a particular production scheme. There is a wide array of expression vectors available for producing protein in E. coli. The most commonly used systems are those derived from the lactose operon, where a strong promoter is under the control of a lac operator. This allows the target gene to be switched on when desired, typically after the biomass production phase, using so-called inducers, most often IPTG, otherwise known as isopropyl-thio-beta-galactoperonoside. Vectors carrying the T7 and T5 promoters are the most popular. There are other expression systems available, but all are based on some sort of controlled expression system. Often it is desirable to fuse the target protein to an affinity tag. This tag can simplify the downstream purification, and in case of the maltose binding protein MAL-E and the glutathione transferase GST and thioredoxin TRX, can sometimes keep the desired protein in a soluble form, thereby improving recovery of active product. In cases where proteins are toxic, need membrane translocation to mature, or have a large number of disulfide bonds, exporting the protein to the periplasm is desirable. Inserting a signal sequence code 5' to the coding sequence of the target protein places an export signal on the amino terminus of the protein, thereby directing it to one of three export pathways. This targets the protein to the periplasmic space and, in rare cases, exported to the culture medium. For historical reasons, many commonly used protocols for the expression of proteins in E. coli employ LB broth culture medium. The LB broths were first developed in the 1950s, have three different versions often called Miller, Lennox, and Luria, after the scientists who devised the formulation. These media were developed at a time before the optimum conditions for cultivating E. coli were known, and long before recombinant proteins were first produced in this organism. These formulations worked for the physiological and genetics experiments of the time. However, the LB broths have a few drawbacks. The LB broths are composed of just three ingredients: yeast extract, casein hydrolysate, and sodium chloride. The difference between them is the amount of sodium chloride, which is 10, 5, and 0.5 grams per liter for the Miller, Lennox, and Luria broths respectively. The first drawback is that these media have no added carbon source and rely on the sugars present in the yeast extract. This can vary from 0.3 to 0.8 grams per liter depending on the source of the yeast extract, and is considerably less than the amount needed for maximum growth. Second, these media are not buffered. As a consequence, during growth there is a buildup of acid, which reduces growth yields, and adding more sugar to these media only exacerbates this problem. Finally, there is no added phosphate, sulfate, or potassium, which are needed in substantially higher levels to achieve maximum growth yields. In essence, the LB broths were not designed or intended for production of recombinant proteins, and in fact in commercial production systems, more nutritionally complete medium is used in the fermentations for the production of recombinant proteins. About 40% of proteins express poorly or not at all in E. coli. In the course of assisting clients over the past 20 years resolve this limitation, we have found that the type of carbon and nitrogen source can make a significant difference as to whether a protein can be produced in sufficient quantities. Using this knowledge, we have devised a set of media that routinely yield more recombinant protein per unit volume than LB broths or more traditional media. In shake flask cultures, as is shown in the table, cell biomass yields can be 2.5 to 3.5-fold higher as compared to LB broth. More importantly, these formulations have been shown to increase the production of recombinant proteins. The SDS page analysis shown are the results of expressing 6 different proteins in each of 6 media, LB broth plus 5 of the special formulations. As can be seen for GFP, TESA, and LYPA, the amount of protein produced is significantly different depending on which medium is used. Importantly, the medium that is best for one protein is not necessarily the best for another. For example, GFP is maximally produced in superior broth and produced well in hyperbroth and glucose M9Y, but does not express well in LB broth, power broth, or turbo broth. In contrast, LYPA is accumulated to higher levels in hyperbroth, followed by power broth, superior broth, glucose M9Y, and turbo broth, and almost no detectable production in LB broth. The difference in accumulation levels is less pronounced for MOL-E, GST, and I27O. Nevertheless, even for MOL-E, a highly expressed protein, the amount accumulated in LB broth is 20 to 30% lower than for the other media. In terms of volumetric productivity, the higher expression levels observed in the specialty media, coupled with higher cell biomass production, results in greater than 5-fold increase in recombinant protein production. These media formulations are also designed to be used in standard fermentation equipment from bench to production scales and can be adopted for autoinduction. A mammalian protein, designated MP8, was produced in an in E. coli in a 400-liter fermenter using different media and expression induced using either IPTG or with lactose feed at the start of the fermentation. The SDS page analysis shows that the amount of protein accumulated in hyperbroth lane 5 and power broth lane 6 using lactose induction was significantly higher than with standard fermentation salts using glucose and IPTG induction. With other proteins, IPTG induction was as effective as lactose was in this particular study. Importantly, not only is more protein made using these specialty media, but in some cases more active protein can be recovered. For example, LYPA, as was shown earlier and in the SDS page analysis shown here, the amount of protein accumulated in all media except LB broth was a significant fraction of total cellular protein. However, when the enzymatic activity was measured in the extracts, as shown in the bar graph, not all media yielded active protein. Microscopic examination of the cells after induction revealed the presence of inclusion bodies. Only in hyperbroth did a significant fraction of the protein accumulate in an active form. By comparison, active TESA, a protein related to LYPA, had the highest level of production of active protein in turbobroth. It is clear that the composition of the medium can have a significant influence on the expression and accumulation of recombinant proteins. Richer and pH-controlled media formulations yield more cell biomass and more recombinant protein as compared to LB broth. One critical observation has been that each protein seems to be unique in its requirement, and it has not been possible to predict in advance which medium will work best for any given protein. Therefore, a simple screen of several media will often be the difference between success and failure in producing a recombinant protein.